This is a production of Cornell University.
All right, thank you so much for such a kind introduction and giving me an opportunity to speak in front of such a wise and esteemed audience of plant scientists of Cornell University. So my research, I was lucky enough to develop my research from my PhD all the way through my position as a group leader. So I'm going to take you through that journey, starting with what really amazed me as a plant scientist is that plants, although they have their preferred habitats, they still manage to grow over a wide range of different habitats, from almost desert-like habitats where there is a high range of daily temperatures, all the way to the tropics where the range of temperatures is very narrow, humidity is very high. And what's also quite interesting is that we as a human species managed to build agricultural systems throughout those different habitats as well. So we've got people that are doing agriculture in Australia, as crazy as it might sound, to Europe and India and Africa. So, but right now, as probably most of you are aware of, our agricultural systems are being challenged by the climate change. And in here, we've got an overview of what are the challenges to the agriculture in general. And you see that a lot of those risks are represented by blue, which is representing the water availability, or rather the lack of water availability. So many times when there is no fresh water available, people are doing irrigation, which in turns is resulting in soil salinization over the long term. So how can we improve or manage that our agricultural systems are resilient against those changes? So one way of doing this is to domesticate species that are already loving the extremes. For example, this desert rose, which in my Photoshop fantasy, I put some very juicy fruits. Normally the fruits of the desert rose are very not very appealing to eat, but maybe with some genetic engineering, we can domesticate them and make them produce something that we as humans are interested in. Another strategy is looking into an intraspecific variation. So the variation that already exists within the species we like to eat or use for other purposes, or exploring the variation within the species that are closely related to the species that we like to eat. So, but before we start to equip our agricultural systems with all of those different mechanisms of resilience, we still need to understand what are actually the plant responses to apply those resilience mechanisms as efficiently as possible with as little yield damage as possible as well. So I'm focusing on the salt stress, and salt is usually occurring in the soil. So the plants, when they are exposed to the salt, the first reaction is to try to pump all of the ions out of the root against the gradient. And as pumping anything against the gradient, it is very energetically expensive and over the long term, not very efficient. So another part of the response is whenever the salt is accumulated in the soil in very patches, the plant will propagate its roots mainly in the portions of the soil that is not that much affected by salinity. And it's known as a negative halotropism, which we described in the Christa's lab with Carlos, Galvan, and Puria. However, at some point, plant needs to take up the water. So it takes up ions and uses them as an osmoticum to be able to increase the hydraulic conductivity into the root and all the way so that the plant can continue transpiring. If the salt concentration is high, at some point the leaves that are the oldest, that have been exposed to salinity for the longest time, they are undergoing a premature senescence and the yield is probably slightly decreased. But if the concentrations of the salt in the soil is reaching the photosynthetic apparatus in the younger leaves, they're also undergoing premature senescence and the yield is basically negligible of those plants. So as you see, this overall salinity tolerance of the plant is pretty complex thing. It's comprised of multiple processes that are compounding on top of each other. So that's why there is this example of the study of Katori et al. From a decade ago right now, where they studied the different accessions of Arabidopsis and looked throughout time when watered with quite high salinity. So this is equivalent of Red Sea salinity. And they looked at how those plants are surviving or not surviving throughout time. And they failed to identify any genetic components because they were looking at salinity tolerance as a whole. Well, when you're trying to map all of those different processes at the same time, you simply don't have enough statistical power to do so. So what I was told very early on in my career is to focus on specific tissues or specific processes that you hypothesize are contributing to salinity tolerance. And I might comment on that later when talking about high throughput phenotyping facility. But without knowing any better, I decided to focus on root system architecture. So when as a PhD student, I started growing those Arabidopsis plants on increasing concentrations of salt, I observed that, yeah, plants, the more salt you add, the smaller they grow. Not very surprising. But the question that I really was excited about addressing is the salt or stress in general only delaying plant development, or is it really reprogramming it? So I scratched my head for a couple of minutes to figure out how are we going to do that. And I decided to look at the dynamics of root development throughout time and under different salt stress conditions. So what we basically did is we germinated the plants under control conditions for 4 days. And at 4 day old, the root, there's just the main roots. There's not much architecture per se to a plant. So at 4 day, we transplanted those seedlings into 0, 75, and 125 millimolar of salt stress. And we scanned those plates, very high-tech technology, scanning of the plates every second day to observe how the plants are developing under those different conditions. And then later on, we also quantified this development using a semi-automatic or semi-manual software called EasyRhizo. So once we had the heaps of data, I had to think about how to synthesize it in a way that I can really make comparisons. So what I did is I set up a very simple model to describe the growth of the root system architecture per se. So I divided it into 3 components. So we're starting with this main root that is just a tiny little root at the 4 days. And that root is growing with a certain speed. And I could fit a quadratic function into that. Oops, sorry. So you've got the starting point, and then you've got the growth rate of the root. At some point, you also have lateral roots popping up. And the speed at which the lateral roots are emerging is also the increase of the lateral root number. So we can also fit similar type of function in there. And then you also have the outgrowth of those lateral roots, which you can then measure with, like fit an average lateral root length speed to it. So basically, you can summarize the entire dynamic of the root system architecture in just 3 numbers. And by adding and multiplying them together, we could definitely predict the total root size of the entire root system, which was really cool. So when I quantified all of those different parameters in the 3 different conditions, for the main root, number of lateral roots, and average lateral root growth, what I observe is that you've got the decrease, the more salt you add, the slower individual components are growing. Again, not very surprising. But because we're using the same kind of family of mathematical formula, so this is a quadratic formula, we can actually compare the differences caused by salt between individual components. So when we calculate the relative change in the main root, number of lateral roots, and average lateral root growth between those components, we observe that in Colombia, which is a reference genotype of Arabidopsis, the main root is impacted much more severely than the number of lateral roots and average lateral root length in both conditions, which was pretty exciting because it means that they are not only growing just slower, they are really growing differently. Because if they would be impacted with exactly the same extent, then we wouldn't see any of the differences in the relative change, right? Okay, so then we quantified multiple accessions. So we classified them, some of them that were responding exactly the same as Colombia. Some of them were having exactly the opposite. So the main root kept on growing, but the lateral roots not so much. So we also performed quantification of the root system architecture on a wider population, which comprised 350 accessions. And for this population, back then when I was starting a PhD, we had a data set of 250,000 SNPs available, which allowed us to do a genome-wide association study. So connecting our different phenotypes of the root system architecture with the SNP markers. So here you see a typical output of the genome-wide association and Manhattan plot, association strength on the y-axis and 5 chromosomes of Arabidopsis. And here is an association that we were most excited, and the association was with average lateral root length. And why were we the most excited about this association? It's basically because it was association with HKT1, which stands for high affinity potassium transporter. So that's the beauty of salt stress field. So in multiple plant species, there are a number of HKT1. In Arabidopsis, there's only one HKT1, and it's not transporting potassium, but it's mainly transporting sodium into the cell. And we were very excited because HKT1 was already previously mapped by Ivan Baxter to be associated with sodium accumulation, trait that we know was very much correlated with salinity tolerance, but it was not previously associated with root development. So we made use of the lines that were available for those HKT1, which were overexpression lines in the xylem pole pericycle, which is a native expression site for HKT1. So why is HKT1 correlated with salinity tolerance when it's actually pumping ions in? So it's the key is where is the native expression of the HKT1. So it's expressed in the xylem pole pericycles, which are the cells that are surrounding the transpiration stream and are basically sucking all of the ions out of the transpiration stream so that sodium is not accumulating to the toxic levels into the shoot. So when we looked at those lines, and the Inge Müller generated those lines in both Colombia and C24 background. So here we've got the background lines and the 2 lines that have an overexpression of HKT1. When we grew them under control conditions, there is not much difference. But as soon as we added salt, we observed that there is the plants are really suffering, and they're mainly suffering in their lateral root development, as you can see demonstrated in here. Well, no significant differences were observed under control conditions. So that was exciting, but then we had to think, like, how is that possible? So HKT1, as I already told you, is expressed around the xylem pole pericycle, around the xylem vessels. And that's exactly the same tissue that is giving origin to the lateral roots. So if you over accumulate those ions in this very tiny, precious little lateral root primordia, they're going to die. So we thought, okay, this is a good hypothesis. Let's test it in another biological system. So there are lines, wheat lines. So wheat has 5 HKT1 genes. And to make it easier, HKT1 that is the most homologous to Arabidopsis one is NAX2 locus. And in here, we've got the introgression lines of another HKT1 and an HKT1 that is homologous to Arabidopsis. So this is the NAX2. And we got those lines from Rana Munz and Richard James from CSIRO in Australia. And we screened them very early on in their development for changes in their seminal root length, lateral root number, and lateral root length under control conditions and salt stress. So for the seminal root length, we didn't really observe that much of a difference, either in control or in salt stress. But when it came to number of lateral roots or average lateral root length, we saw a little bit of a phenotype under control conditions, but it was definitely much more pronounced under salt stress, that those lines with high HKT1 expression in the weed developed more lateral roots, which is exactly the right phenotype in exactly the wrong direction. So we were again scratching back to the drawing board, scratching our heads. How is that possible? And reviewing some basic developmental biology and differences between the monocots and dicots. Well, HKT1 in wheat is still expressed around the xylem. The lateral roots are actually originating from the phloem pole pericycle. So HKT1 is giving much better protection for in the wheat without damaging the lateral root development, unlike in the Arabidopsis. So this is at the moment the working hypothesis that we're dealing with. Another interesting phenomenon that we observe is that when we supplement it, so HKT1 is mainly transporting sodium, but it also transports a little bit potassium. And salt stress is not only about ion accumulation, but also getting a disbalance between sodium and potassium. So we were wondering, if we add a little bit of potassium to the media together with our salts, can we then alleviate this dramatic decrease in lateral root development? And the answer is yes, we can. Although in the background lines, the average lateral root length went down because you've got higher overall osmolarity. We could observe a very small but still significant increase in the lateral root development in the lines overexpressing HKT1. So we thought, okay, let's use HKT1 as a tool to learn more about what's happening to the lateral root development under salt stress. And so we did what many people do when they want to move forward and do a hypothesis generating experiment. We did RNA-seq, where we again germinated those seedlings on the control conditions. We transferred them to 4 different treatments. We let the seedling be there for 24 hours, and then we harvested only the root from all 4 genotypes per experiment. So one sample consisted of 150 to 200 seedlings. So you can imagine I was harvesting forests of Arabidopsis. We had 3 replicates per line per condition, and we only harvested roots. So after doing, sorry, not the right direction. So after doing differential expression for individual line between control and individual treatments, we also did a double differential expression between the background line and the line overexpressing HKT1. So we've got a response not only to stress, but also to HKT1, which is background specific. So after we did that, so here we see differential expression in response to salt and HKT1 and C24 background. And here we've got the same for the Colombia background. And here we've got salt and potassium for C24, salt and potassium for Colombia. And when we looked at the overlapping genes, surprisingly enough, we didn't find that many genes that were overlapping between 2 different backgrounds. But we also know that those 2 different lines are having very fundamentally different responses to stress. So in the end, we were happy to have any overlap, I would say. And when you have little overlap, it's easier to pick the genes that you're going to focus on. So one of the genes that we focused on was shared between Colombia and C24 response to salt and HKT1. And it was encoding TMAC2, which is a negative ABA regulator. And when we looked at the relative expression of this TMAC2 in the Colombia and overexpression line and C24 and its overexpression line, we observed that the pattern was actually, it was significantly changed, but it was exactly the opposite, which was very, very interesting. So we couldn't get our hands on any of the mutants of the TMAC, but we did measure ABA accumulation in collaboration with, Jiangyu Wang from Salim Al-Babili lab in KAUST. And what we observed is the pattern that was exactly complementary to an expression of negative ABA regulator. So whenever our negative ABA regulator was upregulated, the ABA levels were down. Whenever it was downregulated, the ABA levels were up. So there might be some feedback mechanisms between this ABA negative regulator and the ABA biosynthesis itself, which I think is quite interesting. So at the moment, we're generating the CRISPR mutants in all of those background of C24 Colombia and both tissue-specific overexpression lines to see, do we have, can we reverse this decrease in lateral root emergence under salt stress? Another gene that we also explored was TIP2-2, which encodes a tonoplast intrinsic protein, which is supposedly an aquaporin, although nowadays there is an increasing evidence that aquaporins are transporting much more than just water. So over here, we observed that in both lines of overexpression, the TIP2-2 expression was upregulated. And luckily, we could get our hands on TIP2-2 mutant in Colombia background, and we explored their root system architecture under control, salt and more salt conditions for total root length, number of lateral roots, and main root length. And we didn't see that much of a difference under control conditions, but as soon as we added salt, the difference in the total root length was becoming more and more pronounced. And we figured out that it's mainly because the TIP-2-2 mutants developed much more number of lateral roots. So again, right now we're generating the CRISPR mutants in the background of the UAS HKT1 lines and their retrospective background lines. And also we're working in collaboration with Steve Tyerman and Caitlin Bird in Australia with the transport properties of this TIP-2-2 because it hasn't been that well characterized yet. So this was all focusing on the root system architecture. And as a root biologist, I tend to be very proud because I'm working on the hidden part of the hidden half of the plant, but it's still a half. So talking to Mark, we realized that we don't really know what's happening to the root to shoot ratio in salt stress, unlike we know what's happening in the drought. So together with first Rashid Aid Hadou, we developed this tool that was able to transfer our scanned pictures into just green, white, and blue pixels. And it's a semi-manual analysis tool. So I only say it's semi-manual because if you, as a PhD student and postdoc, get to work with semi-automatic, making all of those manual corrections, you realize that it's quite still a lot of manual work. But we still did it. It wasn't perfect because we were still not able to deal with plants where the roots are overlapping. So there might be still a little bit of an error, but we were willing to take that risk of that error and use the tool to calculate the growth rate of the root and the shoot. And here, again, we fitted very simple mathematical functions. And here we're using exponential functions to estimate the root growth and the shoot growth of all 350 accessions of Arabidopsis again. And again, the more stress you add, the slower the plants are growing. But the more interestingly is that when you look at the root to shoot growth rate ratio, you see that it's not really responding in a certain way. So the plants are not getting rootier as they would under drought stress, but actually they are just getting dysregulated. So the root and the shoot growth rate is not as well coordinated as it is under control conditions, but it's just going both ways. So we did genome-wide association, and we used shoot per total seedling area as our indicator of root to shoot ratio, among other things. And one of the associations we found was on chromosome 3, and this association was in the DUF247, which, if you don't know, stands for domain of unknown function 247. So far for creative naming. Anyway, so it was in the valley of other DUF247. And we looked at the sequence diversity across HapMap accessions. We observed that there are some insertions and deletions and that there is some sequence divergence in here. And when we use the newer SNP set containing 10 million SNPs that was provided to us by Arthur Korte, well, the threshold went up, but we found much more SNPs that were still above our previous threshold of 6. So we found around 50 associations, 50 SNPs within this DUF247. So what we also did is we looked at all of the SNPs that were in the coding region of the DUF247 and performed haplotype analysis. So we grouped now the accessions not per SNP, but just per the clusters of SNPs. And we found that cluster 11 was significantly shootier than the cluster 1, which contained our favorite Columbia 0. So when we started to clone those different alleles together with Mashel Al-Katani, who is a PhD student in Mark's lab, we observed that there are actually 2 huge 200 base pairs insertions in here. And they are leading to a premature stop codon, which is basically resulting in lack of the DUF domain. So we also know that there are 26 other DUF247 in Arabidopsis genomes. The closest similarity are the DUF247 that are within the same genomic region, but they are showing very little expression similarity. So right now in the lab at the BTI, Haley Sussman is working on the functional characterization of the different tDNA insertion mutants of not only the DUF that we're interested in, but also all of the other ones that are in the neighboring regions, and also figuring out where is this DUF247 localizing to. Liang is looking at the evolutionary context of the DUF247 in collaboration, of course, with Andrew Nelson. And Anna Nelson is doing molecular characterization of the DUF247, looking at calcium binding, dimerization, and also at those characteristics of the domains. So now for the whatever time I've got remaining, I want to kind of switch gears and talk about, like, do we really need to focus on individual traits? So traditionally, as a biologist, we learn to use forward or reverse genetics and to identify the relationship between a gene and a phenotype, or a phenotype and an allelic variation within a gene in terms of forward genetics. And there's absolutely nothing wrong with that. And we learned a lot. We learned which processes are important for salinity tolerance, which genes are, among other genes, contributing to that, how are all of those different genes interacting. But what we're still missing from one picture is all of the other traits that could potentially contribute, that we are not even hypothesizing are contributing. So this is, I think, where high throughput phenotyping is coming in, and high throughput enabled discovery, because then you can think about your the interesting process that is of your interest, and without the need to focus on only one phenotype that you can score. So you can score multiple phenes. So those are basically a relation to a phene to a phenotype is the same as gene to a genotype. So it's very elemental function of the phenotype. You can score them throughout time, and the denser you go, you can also observe very transient processes which are still occurring, but we're very often missing when we're doing it in the field or when we're doing phenotyping that is destructive. So we can not only quantify how the phene contributes to overall plant performance, but also what is the individual contribution of the phene to each other and how they are reacting and how those interactions are changing throughout time and throughout different conditions. So I've got some papers that are already, where we're looking into how can we use high throughput phenotyping into gaining more insight about the process in general, looking beyond one phenotype and beyond one gene. So one example is this paper that was published with Gigi Gao in Plant Phenomics, where we basically took Colombia and a mutant that we know is heat sensitive, and we phenotyped it with the high throughput facility, and we developed a logistic regression model where we trained it on the subset of our data, and then the model was able to distinguish whether a plant was a mutant or a wild type based only on the inputted phenotypes, and then we could retest it on the remaining set of the data. And usually machine learning is being seen as this black box, but I'm really interested to look what's inside and making that black box transparent to see which of the traits are actually used by this logistic regression with more seriousness, are being taken more seriously into consideration how to make a decision. And like this, we can also learn which of the traits are contributing to this distinction between mutant or wild type, or stress and non-stress plants and things like that. And I think that this might be a powerful approach later on. Another paper that is currently under revision, but you can find the bioRxiv version of it, is a paper that we worked together with Mariam Aulia, who focused on early responses to salt stress. So she developed a protocol to apply salt stress in the soil and then phenotype the plants for the subsequent 7 days. So we end up with this big data tsunami, which we then used for multivariate analysis and machine learning. And in this case, we didn't use logistic regressions, but we used tree-based models. And that allowed us to identify the traits that have a high contribution to overall plant size under control and salt stress conditions, and also took that information when we were reviewing the genome-wide association outputs. So we knew which of the traits we should give a little bit more love to and attention. So that led us to identification of this uncharacterized protein kinase, which affects quantum yield under light adapted conditions. And then also the mutants showed a significant differences with Colombia for just overall plant area and plant growth, which I think is pretty exciting. And I don't think we would have focused on one of the 700 associations that we found without doing this step. All right, I don't really have that much time to talk about that, but I also was involved in developing multiple tools for dealing with high amount of data. So MVApp is one of the results where it allows you to do quick pre-processing of your data, identify outliers, which usually takes quite a lot of time, but also evaluate the traits for the broad sense heritability, do some curve fitting so you can estimate the growth rate, summary statistics, but also hypothesis testing. So we usually do ANOVA, but nobody really does testing assumptions for ANOVA. So it's there, you don't have any excuse now. But also a lot of hypothesis generating tools, which is quantile regression, clustering, correlation analysis, PCA. So all of this is based on R, but we just package it into a user-friendly, clickable approach. And there's also a YouTube channel and a Twitter account where you can find quite a lot of resources on how to use that. We're dedicated in maintaining this tool and keeping it updated based on extremely positive feedback from the community. So just quickly, what I want to do at the BTI during my appointment as an assistant professor in here, I want to focus on species that are naturally tolerant to stress. And one of them is Solanum pimpinellifolium, which is the closest relative of tomato. It's native to the west coast of South America. And it's super tolerant because it survived in my garden in Saudi Arabia. So I think that's a very good argument for working on something. I'm a very poor gardener. So Faye from the BTI, they recently released the reference genome for the pimpinellifolium, and we at KAUST sequenced 230 accessions, which we're right now processing for the SNP calling and making it as a useful genome-wide association resource. So what we want to do with this population is going to be our guinea pig together with Andrew Nelson, which I was hired together with Andrew as a cluster hire. You can read all about it on the BTI website. I'm not going to talk that much, but what we want to do is we want to phenotype this population using traditional high throughput phenotyping, so scoring growth, chlorophyll fluorescence, doing multispectral imaging, things like that, but also using Raspberry Pi phenotyping, which will allow us to have much higher time density. So look at the changes that are occurring within minutes of the plant, and that will allow us to identify things that are very often overlooked, like quiescent phase that plants undergoing after stress exposure, and also how quickly the plants recover and what are the dynamics of this recovery. So those high quality changes in plant growth will allow us to select time points at which we're going to collect more molecular phenotypes, such as transcriptomics, metabolomics, epigenomics. And using all of this data, we're going to be able to perform basically like GWAS on steroids, which is not only association between your phenotype and the SNP, but also your phenotype and the transcript levels or metabolome levels. And then this layered approach is going to allow us to see which of the associations are very stable and confirmed by different layers, and also pick the ones that we're the most excited about. In the larger scheme of things, we have a very evil plan on exploring not only pimpinellifolium, but closely related cultivated tomato, and then also branch out to all the brassicaceae, and later on also to monocots. And this comparative framework will allow us to establish which of the resilience mechanisms are very short range, applied and only found within the species, medium range, so applied and found within the closely related species, and long range. And that kind of framework will allow us to see what are the mechanisms that are easily transferable between the species, and what are the mechanisms that are more species specific. Yeah, and in the end, I wanted to thank you for your attention, but also my group that I did a PhD. So this is Krista Testering Labs back at University of Amsterdam. Krista right now moved on to Wageningen University. She was an excellent mentor and gave me quite a lot of freedom in my PhD. Then I did a postdoc in Mark Tester's lab. So here is basically a view from the lab. We're at the Red Sea coast. So Mark was also an excellent mentor, and all of those amazing people were great colleagues and contributors. Oops, sorry. And also all of the collaborators and colleagues, students from KAUST. And of course, last but not least, I wanted to thank all of my new colleagues here at the BTI, who are a wonderful team to work with. So Haley and Liang, and of course, Andrew and Anna, who were absolutely instrumental when I was starting here, just clueless about where everything was, who was doing what. And also the BTI senior leadership team, who managed to get me from Saudi Arabia to Ithaca, from all the places during this weird time of COVID. And that's it. I would like to welcome all the questions. After a presentation, it's always like, is anyone else there still?
You can unmute yourselves and ask a question, or you can just put it in chat.
So Magdalena, this is Wojtek. So in your GWAS on steroids, you mentioned, and I saw it in the slide, that you want to take the environment into account as well. And I wonder, like, if you could elaborate on that, like, I'm curious, like, how and you're going to do it in practice.
Yeah, so the environmental GWAS, it's not our idea. I think it originated from Ed Buckler, from over the road. So what we would be doing then in environmental GWAS is collecting the GPS coordinates of all of the accessions where they are originating in. And then you use the environmental traits, so annual precipitation, temperature, stuff like that, as your phenotypic traits. So that would then, if you've got an overlapping association between your phenotype and this environmental GWAS, it would say, oh, this is something that is causing local adaptation to this specific climate, to this specific environmental variable. Does that answer your question, Wojtek?
Yeah, sure. And so that then you'd have to like pay attention to like geographical distribution of the whatever lines or accessions you're collecting, right?
You would no, well, so you would always have a kinship matrix as your cofactor in your GWAS.
Yeah.
That would not exclude the need
Sure.
for that, because running that as a cofactor or running it as a separate genome association is very, very different.
Yeah.
But what would so we would be able to do that. But I also realized, based on the work that I've done on Arabidopsis, and a lot of people are like, oh, all of those accessions are from the coast, and therefore they must be more affected by salt. Well, salt is a stress that is very difficult to make this kind of assumptions based on just global data, because it's very depending on the type of the soil that you've got and things like that. I would assume that the west coast of South America would be less disturbed by that, but I don't know. I think it would be still good to do it, especially with the stresses that we're planning to work on. So not only salt, but also heat stress and drought stress. So those parameters are much, much easier to estimate from the environment and the data that is available.
Thanks.
Other questions, anyone? Okay, I see nothing in chat. Okay, let's thank Magdalena for a fantastic seminar.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.